Hello everyone, welcome to Black Screen. My name is Aksh Patel and I'm a third year medical student from NHLMMC Ahmedabad. Today we will be looking into a very important question from university exam point of view, and that is culture media. So let us start culture media. So before dealing into brief of culture media, let me tell you that we will be studying culture media under three headings, and those are: first is definition, second is constituents, like of what all material the culture media is made up of, and the third one is types of culture media, and this is a very important topic. Okay, so let us start. What is the definition of culture media? So culture media is nothing but liquid or solid substance that contains nutrients to support growth and survival of microorganisms. So this is the theoretical knowledge. Actual meaning is culture media is a media that helps microorganisms to grow. It contains several favorable conditions, favorable atmosphere, favorable nutrients that helps bacteria, fungus, virus to grow. That's simple. Now, what all are the constituents of a culture media? Like culture media is made up of what materials? So there are more than 100 constituents, but what is needed to be remembered are some important 3, 4, or maybe 5, 6 constituents that are frequently used. Like they are the universally used. Okay. The first thing is water. Culture media contains water. Then we have peptone. Now, what is peptone? So peptone is partially digested protein. The third one is blood. Then we have yeast. Then we have meat. Okay. So these five constituents are most common. We also have some other things, but whole bunch of list doesn't has to be remembered. Okay. Now, the most important thing, which is also separately asked as a short note, is types of culture media. So we can divide culture media into two main classification. That is classification based on consistency and classification based on properties. Okay. Now classification based on consistency. Now you all know what consistency is. We can divide culture media into solid and liquid. Okay. So we have solid media and we have liquid media. Now remember, one important thing is in microbiology we call liquid media as broth and we call solid media as agar. So let me write in bracket, we call liquid as broth and solid as agar. Examples of liquid media is anything which you come across in microbiology that has the name broth in the suffix, or I mean the second word. Okay, so examples are nutrient broth. Then we have alkaline peptone broth. And examples of solid media are nutrient agar, blood agar, chocolate agar, anything which has the name agar at the back. Okay. Now, if I talk about the classification based on properties, then we can have further two sub classification, and they are based on, sorry, based on conventional methods, and we have automatic methods. So we classify culture media based on conventional media, and we have automatic media. Now this conventional media are actually seven. We further subclassify conventional media under seven subheadings. And automatic, we have two, three. You can remember as much as you can, but I recommend you to remember at least two, because this is asked as a whole short note. So if you write each and everything in detail, then you will fill more than 7-8 pages, which is not required. So we'll write everything in brief and superficial. Okay, so now let us start conventional media. Okay, so based on properties, I've told you the culture, conventional and automatic, and in conventional we have seven headings. So these are simple, enriched, selective, enrichment, differential, anaerobic, and transport. You can remember these seven names by a short note, by a mnemonic. Sorry, by a mnemonic called SE square, because you have SE, simple, enriched, selective, enrichment. And then you can remember as DANT, like DANT. So DNT, DANT. So differential, anaerobic, transport. And you can remember automatic as backtech and bacti alert. Okay. So now this conventional media, they are also asked separately as a 6 marker short note, in which you have to explain the conventional method of classifying the cultural media. So now let us see each one into detail. Okay, so starting with the first conventional media, that is simple media. Name itself suggests this is a simple media, that it doesn't contain any extraordinary thing. We also call this simple media as basal media. Let me write over here, this is basal media. Okay, now this simple media helps the growth of non-fastidious non-fastidious organism. Now, what is non-fastidious organism? So, any organism that doesn't require any special nutrient for its growth. Special nutrient in the sense, some microorganisms require blood, some may require egg, some may require meat. But these are the organisms that doesn't require any special like nutrients to grow. Exact opposite is fastidious organisms. Now, these are non-fastidious. If I talk about the examples, they are Staphylococcus. Okay, sorry. Then we have Enterobacteriaceae. Okay, so these are non-fastidious organisms. Now, example of simple media includes peptone water, peptone broth, and nutrient agar. Let me write peptone water, peptone broth, and nutrient agar. You can remember PPN, peptone water, peptone broth, and nutrient agar. So this is the simple media. Now let us write about enriched media. Enriched media. Now, how will you define enriched media? So, enriched media is a media that is prepared from simple media by adding special nutrients. Like as I've told you in non-fastidious organism that it doesn't require anything special. Enriched media is a media that is prepared from simple media by adding something else. Now, what something else mean? That is blood, egg, meat. So it helps in the growth of fastidious organism. Okay. So this enriched media is nothing but simple media plus extra nutrients. Okay. It helps in the growth of fastidious organism. Okay. Now, if I talk about the examples of enriched media, then we have blood agar, chocolate agar, LJ media. You can remember any two. Now, let me write the examples first. So we have blood agar, we have chocolate agar, we have LJ media. Now, how these are prepared? Like as I've told you, this enriched media are made using simple media by adding some special things. So how this blood agar is made? So blood agar is made from nutrient agar, which is a simple agar, like simple media. So this is made using nutrient agar plus adding blood to it. Now how chocolate media is prepared? Same. We have nutrient agar and then we add blood. Now what is the difference between chocolate agar and blood agar? Like the process of making seems to be the same. But the only difference is first you heat nutrient agar to 50 degrees Celsius, 50 degrees Celsius in blood agar followed by adding blood. And then in chocolate agar you have to heat nutrient agar to 70 degrees Celsius followed by adding blood. Okay. Now in LG media we have nutrient agar plus egg. Simple. Now let us quickly look into enriched media. So enriched media is made from simple media by adding some extra things. It helps in the growth of fastidious organism. Then the examples are blood agar, chocolate agar, enriched, LJ medium. Okay. Now let us move to the third thing, and that is selective media. Okay, so let us start the third one, that is selective media. Now, what is selective media? So selective media is a media that allows the growth of a particular organism and inhibits the growth of rest all organisms. Like for example, if a physician suspects a patient of cholera, so what microbiologist will do is uh, he'll make a particular media that will allow the growth of Vibrio cholerae. At the same time, it will also inhibit the growth of all other bacteria. Like for example, it can be Staphylococcus, Enterobacteriaceae. These are not the organisms of our interest, so we want all those to be inhibited. Okay. So selective media is a media that allows the growth of a particular organism of our interest. At the same time, it inhibits all other organisms. So this is what I've written here. Now, as I've said you in enriched media, we add something extra nutrient to the simple media. In the same way, in selective media, we have a thing called selective agent. This agent When added to selective media, we have growth of a particular organism and inhibition of rest all organism. Okay, so let me make a table that will help you understand this better. Okay, so we have this thing called selective agent. Then we have when the selective agent is added to a media, that is selective media. So that also has to be added to the heading. So this is selective media, and when this selective agent is added to selective media, it helps in the growth of which bacteria, which is the bacteria of our interest. So that also has to be added to the heading. So the third heading is of selective for. Selective for what? Okay, so this is selective for. Okay, now let us start the first selective agent. Like there are ample amount of examples given in Apurva Shastri, but only a few things you have to remember, like 2 or 3 examples. The first thing, the first selective agent is pH. So when we add, like when we set pH of a particular media in a particular range, it will allow the growth of only a few types of bacteria, like 1 or 2 bacteria, and it will inhibit all other. So for example, when we add pH, like when we set pH between 8.2 to 8.4 in a media called TCBS media, it will allow the growth of only Vibrios. Okay. The second example is antibiotic. So when we add penicillin as a selective agent to a media called PPLO, that is plural pneumonia like organism media, that is PPLO media. So it allows the growth of only mycoplasma. Sorry. So it allows the growth of only mycoplasma. Why so? You don't have to remember why so, but let me tell you as a fact. So mycoplasma doesn't have cell wall, and penicillin acts on the cell wall. So since this mycoplasma doesn't have any cell wall, how can penicillin act on mycoplasma? So this is resistant. So this is resistant. Okay. So this is resistant. So when we add penicillin as a selective agent in a media called PPLO, it allows the growth of only mycoplasma and it inhibits rest all. Okay. So you can make SH table in the university exam. So let us quickly summarize what is selective media. So selective media is a media that grows a particular bacteria and inhibits rest all. Examples are selective agent like pH, penicillin when added to TCBS and PPLO respectively. It helps in the growth of Vibrios, mycoplasma respectively. Okay. Okay, so now let us move to the fourth media, that is enrichment media. Now, before moving to the fourth media, I have forgot to tell you one thing about selective media is these medias, all the selective media are solid in nature. All the selective media are solid in nature. This is exactly opposite of what it is in the enrichment media. The fifth one, enrichment media is in liquid. The concept is same. It allows growth of one organism and it inhibits the growth of restol. Now directly jump to the examples. Examples includes alkaline peptone water. It helps in the growth of vibrios. The second is selenite F broth. Broth means liquid. Okay, it helps in the growth of salmonella. You can remember it as SS, selenite salmonella. Now the fifth media, that is differential media. Let us quickly go to the differential media. Differential media. So differential media is a media that differentiate groups of bacteria into various subgroups based on colony forming. So if I talk about differential media in simple words, so let us take one example. We have hemolytic bacteria. So hemolytic bacteria is a group of bacteria. It can be further divided into alpha hemolytic, beta hemolytic, gamma hemolytic. The differential media is a media that differentiates hemolytic bacteria into three: alpha, beta, gamma, by making different colors for all three. Got the point? Like we have, let me tell you one example, that is blood agar. Blood agar is, which is enriched media, is also a differential media. How? I'll tell you. So blood agar differentiates hemolytic bacteria into alpha hemolytic, beta hemolytic, and gamma hemolytic. Uh, you can refer Apurva Shastri for examples of beta hemolytic. Uh, if all the hemolytic bacteria are present in a culture media, then it will divide by making different colors for each one of them. Like for alpha hemolytic, it will make yellow color. This will have green color, and when this is present, it will have no color change. Like if blood agar, sorry, this is blood. Okay, so blood agar is red, it will stay red. So no color change. Okay, what is differential media? It helps to divide a group of bacteria into various subgroups. Let us see another example, that is McConkey agar. We have McConkey, agar. Okay, what is McConkey agar? McConkey agar divides gram negative bacteria. So gram negative is a very huge group. Gram negative bacteria into lactose fermenting and non-lactose fermenting. So those microorganisms that are fermenting lactose will give one color, and those who are not fermenting will give another color. So this is differential media. Now let us quickly go to the another media, that is transport media. Okay, so transport media, as the name suggests, transport means it helps in transporting a particular specimen from one place to another place without changing its form, without allowing commensals. Now I've told you what commensal is. Commensal is any organism which is not of our interest. So if we are transporting Vibrio cholerae, a media containing Vibrio cholerae from one place to another place, we don't want Staphylococcus to grow in that media. Okay, so that are commensals. So transport media, that is the sixth one. Transport media is a media that is used to transport uh, like culture media containing specimens from one place to another place. You can make your own definition. That is very simple. The main thing that has to be remembered are the examples, because this transport media, it was asked once in our university for 2 marker, like write any 4 examples of transport media. So let us quickly jump into the examples. So you can remember example of transport media as PV Sindhu. This is the mnemonic which I remembered. So this is PV Sindhu. Okay. So P stands for Pike's medium. This is used to transport S pyogen, Streptococcus pyogen. Okay. Another one is VR medium. VR is Venkatesh Ramakrishnan. You don't have to remember the full form, just VR medium. That is used to transport Vibrio. You can remember it as VV. VR Vibrio. Okay. And Sindhu, S stands for Stewart's medium, and that is used to transport Neisseria. Okay. The fourth example which you can remember, if it is asked for 2 marker, then you have to write fourth examples. Otherwise, these 3 are enough. So if it is asked for 2 marker, you can remember one thing as one more example as Carey Blair. Carey Blair medium. Carey Blair is used to transport stools. Now, this stool contains many pathogenic organisms which are responsible for causing diarrhea, dysentery, etc. So in general, you can write this media is used to transport stools. Okay. Now, last but not the least is anaerobic media. Seventh is anaerobic media. Now, what is anaerobic media? Of course, the name suggests anaerobic media is a media that maintains zero oxygen. It maintains anaerobic media without oxygen, no oxygen. Now there are several agents that are used, like just like the selective media, we have selective agents. In anaerobic media, we have anaerobic agents. So these are the agents that helps to create a condition of zero oxygen. What are those agents? So let us move. So these are the agents, like for example, we have vitamin C. Then we have unsaturated fatty acid. Okay, so these are the agents that causes um, anaerobic conditions. Uh, we also need to know the examples of anaerobic microorganisms. These are also asked as a short question. So let us quickly see anaerobic organisms. They are lactobacillus, then we have clostridium, then we have actinomyocytes, Okay, so these are the anaerobic organisms. Now, the main example of anaerobic media is Robertson's broth. You just have to remember this one example, Robertson's broth. Example is Robertson's, you can remember cook broth. Robertson's broth. Okay, so with this, we come to an end of classifying conventional culture media into 7 subcategories. Uh, if you want to take the PDF of this SH notes, it is available on Telegram channel of Black Screen. You can take. I hope you understand this video. Thank you very much.